Hi, this is Dr. James Hutcherson, and today we'll be talking about the muscular system. And this particular video will start chapter 9, and we'll cover about halfway through it. We'll go through really the general overview of how muscles work, and we'll talk about muscle cells. And then I'll do another recording where we will look at each separate muscle and talk about how they work. So let's go ahead and get started with this chapter. And I'm going to share the PowerPoint screen with you. All right. Well, as you already know from the chapter on tissues, that there are three types of muscle tissues in the muscular system. We've got skeletal muscle, and that's the kind you normally think of because it's attached to bones and moves you around. It is voluntary, and it's under conscious control. Cardiac muscle makes up the wall of the heart. It is involuntary, non-conscious control, and it's responsible for the pumping action of the heart. And then you have smooth muscle that's found in the walls of the internal organs, like your digestive tract, like your intestines. These are involuntary also and under non-conscious control. So we'll start with skeletal muscle, and that's what we're going to talk about most in here anyway, this particular chapter. As we move into like digestive system in Bio 169, we'll go into a lot more detail on the smooth muscle. And then as we move into talking about the cardiovascular system, we'll talk more about cardiac muscle. So there's over 600 skeletal muscles in your body, and they are attached to your bones and also the skin of your face. Again, they're under conscious control, so they're voluntary. They are organs of the muscular system. And as you go back to that first chapter, you'll remember that an organ is a group of tissues that works together for a common function. Okay, skeletal muscles are composed of skeletal muscle tissue, as you would think, but also nervous tissue and blood and connective tissue. The connective tissue part of the skeletal muscle includes the fascia. This is a thin covering of connective tissue around the muscle. And then tendons. These are core-like masses of connective tissue that connect the muscle to a bone. And aponeurosis, which is a sheet-like mass of connective tissue that connects the muscle to bone, skin, or another muscle. The connective tissue in and closely surrounding the muscle includes these three levels or layers: the epimysium, which surrounds the whole muscle, so it lies beneath the fascia; the paramysium, which surrounds the fascicles within a muscle; and the endomysium, which surrounds the muscle fibers, the individual cells within the fascicle. Okay, and this shows that organization we were just talking about. If we start at the largest level, here's the entire muscle. Notice it's tied with the tendon to the bone, and then covering it, we have the muscular fascia. Okay, and then that first covering of the entire muscle is epimysium. Okay, and then we can pull out this entire segment here, which is a fascicle, and notice that the fascicle is covered with the paramysium that's around it. Okay, and then we can go further and pull out the individual muscle fibers, and notice they are surrounded by endomysium. Now we can go even further as we look into the individual filaments or the individual cells. We're going to talk about this quite a bit in this section, how you have a sarcoplasmic reticulum, which covers that fiber. We'll talk about what the purpose of that is. It's just like endoplasmic reticulum in a normal cell. And then you have these individual subunits that are called myofibrils, which we can break down even further into filaments. Okay, so remember each of these levels. Don't get them confused. So when we talk about muscles, we will often talk about compartments in fascia. A compartment is a space that contains the group of muscles and the blood vessels and the nerves, and it is enclosed by the fascia. Now, you may have heard of individuals having compartment syndrome before. So fluid accumulates in the compartment, results in increased pressure in the compartment, can lead to deficiencies of oxygen and nutrients, and causes severe pain. The fascia of each muscle is part of a network of fascia. A portion surrounding the muscle is a deep fascia, and this connects to the subcutaneous fascia under the skin. The network also connects to subserous fascia of the serous membrane. Let's talk about the individual skeletal muscle fibers, or the cell. You'll recall from the chapter on tissues that they are multinucleated. You do have a cell membrane that covers it. We call it a sarcolemma. In other cells, we often just call it a plasmalemma. Then the skeletal muscle cell fiber does have cytoplasm that we specifically call sarcoplasm. And then you have many myofibrils within the muscle fiber, as you saw in that earlier picture. These are long parallel structures. They run down the fiber. They consist of thin actin filaments and thick myosin filaments. And we have a unit that we considered a sarcomere. So these are those units that connect end to end to make up the myofibrils. We'll show you more pictures of these. The sarcoplasmic reticulum is basically the endoplasmic reticulum of muscle, and it stores calcium. And we'll talk about how important that is later. Transverse tubules also, or T-tubules, they relay the electrical impulses to the sarcoplasmic reticulum. And then you have triads, which are units that consist of a tubule and two SR cisternae, or two sarcoplasmic reticulum cisternae. Okay, so this is the close-up. This is a muscle fiber. And again, notice we have these individual units of the muscle fiber. Okay, covering each of those units, again, we do have the sarcoplasmic reticulum, and you have the transverse tubule that covers the sarcoplasmic reticulum. And we'll talk more about that later on. Making up each of these individual myofibrils are, as we showed in that earlier picture, individual filaments, and they are composed of thick and thin filaments, which we'll talk quite a bit more about also. Okay, the myofibrils, they consist of sarcomeres connected end to end. And this causes the striation pattern that we talk about in the tissues. We always say one way you know it's a skeletal muscle fiber is that it is striated. So you see little lines going across it. And it's made from the arrangement of the myofilaments and the myofibrils. Now we can actually divide the sarcomere up into different structures based on intensities of color. The I band, this is a very light band. It's composed just of the thin filaments, which are actin. Then you have an A band, which are darker bands, and they're composed of thick myosin filaments with portions overlapped with the thin actin filaments. Then you have an H zone. This is the center of the A band, composed of thick myosin filaments. You have a Z line or Z disc. This is the sarcomere boundary. It's in the center of an I band. It anchors the filaments in place. And then you have an M line, the center of the sarcomere, an A band, and it anchors the thick filaments. Okay, so this shows you exactly what we were just talking about and what we defined. And again, they're based on how many filaments are crossing each other. So the more filaments you have, especially if they're thicker, they're going to be a darker band that you see here. Okay, and again, we say that we have these individual units of sarcomeres. So here's a sarcomere, and here's a sarcomere, and they just go end to end, on and on and on. This is what it would look like under a microscope, one that has really high magnification. Now, it wouldn't always look this color naturally. Okay, so we've actually added certain dyes and things, but it does give you an idea of why you have certain colors and certain intensities. Like again, you've got the thick filaments here, so that is normally going to be darker, and then you have just thin filaments here, and then you have lines where those thin filaments come into contact with each other at the ends. Okay, and speaking of thick and thin filaments, the thick filaments are composed of myosin protein. The heads form cross bridges with the thin filaments. The thin filaments are composed of actin protein. They're associated with troponin and tropomyosin, and these prevent cross bridge formation when the muscle is not contracting. Now, let me encourage you after watching this for you to go and watch the video that shows contraction of muscles. It'll make a heck of a lot more sense to you if you do that. So here's basically the arrangement. Here we have the thin filaments, again, that's actin, and then the thick filaments are the myosin. And the way that skeletal muscle contracts is that these myosin heads of the thick myosin filaments reach up and grab the thin filaments and pull them. And on this side, they pull them this way. So they move the thin filaments towards each other. And that's what's causing the muscle to shorten and allows you to move a bone. Okay, the contraction of the skeletal muscle fiber requires interaction from several chemical and cellular components. It results from a movement within the myofibrils in which the actin and myosin filaments will slide past each other, shortening the sarcomeres. The muscle fiber shortens and pulls on the attachment points. Now, in order for all of this to get started, we actually have to send a signal, of course, to the muscle somehow. And this is where the neuromuscular junction comes into play. It is a type of synapse. It's also called a myoneural junction. It's where the axon of a motor neuron and a skeletal muscle fiber come together, where they interact. The skeletal muscle fibers then contract when they're stimulated by the motor neuron. So this is the connection between the nervous system and the muscle. Now, look in more detail at the parts of the neuromuscular junction. You have the motor neuron. This is a neuron that controls skeletal muscle fibers. Then you have a motor end plate. This is a specialized folded portion of the skeletal muscle fiber where the fiber binds to a chemical called the neurotransmitter. And it's that chemical being sent from the nervous system of the neurotransmitter which causes ultimately the contraction of the muscle. But now that neurotransmitter has to go across a synaptic cleft. This is a space between the neuron and the muscle fiber across which the neurotransmitter travels. And the way that they travel is through synaptic vesicles. So these are membrane-bound sacs that contain the neurotransmitter. The neurotransmitters are chemicals released by the motor neuron, so from the nervous system, to deliver a message to the muscle fiber so it will contract. Okay, so in this picture, you see the motor neuron axon. Okay, you see the neuromuscular junctions here. This is the synaptic cleft where it comes into contact with the actual muscle fiber. And that's the motor end plate of the muscle fiber. Now, if we look at it much closer, you'll see that there are synaptic vesicles in here that release the neurotransmitter, which then goes across the synaptic cleft to the motor end plate of the muscle fiber. The neurotransmitter we talk about most of the time is acetylcholine, or ACH for short. Nerve impulses will cause release of the acetylcholine from the synaptic vesicles. The acetylcholine binds to the acetylcholine receptors on the motor end plate of the muscle fiber. The acetylcholine causes changes in membrane permeability to sodium and potassium ions, which then generates a muscle impulse or the action potential. Okay, so what the acetylcholine does again is to cause the muscle fiber membrane to become more permeable, so sodium and potassium will move across it. So the sodium and potassium are really what's going to cause the contraction at this point. The impulse causes a release of calcium ions then deeper inside from the sarcoplasmic reticulum, which then leads to muscle contraction. So there's a whole series of events here. And this is one of the reasons why I tell you to watch the videos that are in the course. Okay, so let's talk about a clinical application of this. Myasthenia gravis. It's an autoimmune disorder. Antibodies attack the acetylcholine receptors on the skeletal muscle fibers, the motor end plates, in the neuromuscular junctions. Persons may have only 1/3 the normal number of acetylcholine receptors. So this leads to widespread muscle weakness and muscle fatigue. There are treatments for it. There's drugs that inhibit acetylcholinesterase. That's an enzyme that breaks down acetylcholine normally. There's also immunosuppressant drugs. You can administer antibodies that inactivate the harmful antibodies. And then there's plasma exchange. Okay, some other disorders. Muscular dystrophy. I know that's one you've heard of. It's a deficiency or an abnormality or mutation in scarce muscle proteins that are called dystrophin. Dystrophin binds to the internal side of muscle cell membranes, and it holds them together during contraction. In the absence of normal dystrophin, the cells will lose their normal structure and they'll die. Okay, there's also infection called the Clostridium botulinum. It's a bacterium, an anaerobic bacterium that produces a toxin that causes food poisoning. This grows in food that's not properly preserved. And if a person takes it in, it causes digestive, muscular, and respiratory symptoms. We can treat it with an antitoxin, and it's used in small doses as Botox to smooth wrinkles and to treat migraines. You may have heard of those treatments before. Okay, so let's understand what happens when the acetylcholine binds with the motor end plate and it causes the sodium and potassium to rush in. Okay, because the next part really is the calcium being released from the sarcoplasmic reticulum. So normally during muscle relaxation, calcium ions are stored in the sarcoplasmic reticulum. And troponin and tropomyosin, which are particular kinds of molecules that cover the thick and thin filaments, complex cover binding the sites on the actin filaments. Okay, and these binding sites are where the heads of myosin can reach up and grab the actin and pull them together. Okay, but if the troponin and tropomyosin are covering those sites, no contractions happening because the heads cannot reach up and grab it. Now, when you do have a muscle stimulation, the muscle impulses cause the sarcoplasmic reticulum to release the calcium ions in the cytosol. Okay, so now we're inside the muscle fiber. Calcium ions will bind to troponin to change its shape. And then each tropomyosin is held in place by a troponin molecule. Okay, so what happens then is you get a change in the shape of the troponin, altering the position of the tropomyosin. The binding sites on actin then are now exposed, and now the myosin heads can bind to the actin, which causes a cross bridge. Okay, so let's look at this process here, and we'll start here with the relaxed muscle. Okay, and actually the next few slides go into each individual part of this, but this one slide shows you the entire process. So let's look at it individually. Oops. Oh, it didn't exactly do what I thought it was going to do. So I guess I'll just have to talk about it in that one slide. I don't know if they've changed the PowerPoints over time or what. Okay, so number one, we have the relaxed muscle. So you're actively transporting calcium into the sarcoplasmic reticulum, and this requires ATP. and it makes the binding sites unavailable. Okay, so notice the tropomyosin, which is this long structure, and troponin are these individual structures here. Notice the troponin covers the tropomyosin. Again, when all that's in place, these heads of the thick filament, which is myosin, cannot reach and grab the thin filament and pull it. Okay, now, at some point, as we have a stimulus, ATP is going to cause calcium to be released, and we'll get our muscle contraction. So the release of the calcium from the sarcoplasmic reticulum will expose the binding sites on the thin filament. So notice what happens here. Okay, so we've moved the troponin, which moves tropomyosin, and now the binding sites are available for the myosin head to reach up and grab it. So the exposed binding sites on the actin will allow the muscle contraction cycle to occur. Now the myosin heads reach up, they bind to the actin. That's the cross bridge. Next, the ATP, which had already been there, having been split, the ADP and the phosphate from that will release from the myosin, and the cross bridge pulls the thin filaments. Then we totally release even the split ATP, and a new ATP binds. When the new ATP binds, this breaks the connection, and it recocks the head back in the other direction. Okay, the whole process all starts over again, where ATP causes calcium to be released, which then causes the troponin tropomyosin to be removed, and then the ATP splits, which allows the heads to go up and grab the actin thin filament. When the ATP, which has been split, is completely split, this causes the head to pivot. Okay, so it's like the action on a trigger of, say, a gun. So now that the trigger's pulled, and the hammer now is able to move and pull that thin filament. But the process goes on and on and on. And really what's happening is all these little heads are pulling, not all at the same time. And it's almost like fingers that are constantly kind of wiggling and moving those thin filaments towards each other until ultimately they are fully contracted. Okay, now when the neural stimulation of the muscle fiber stops, acetylcholinesterase, which is an enzyme, will decompose the acetylcholine at the synapse. The muscle impulse will stop when the acetylcholine is decomposed. The stimulus to the sarcolemma and the muscle fiber membranes cease. The calcium pump then moves the calcium back into the sarcoplasmic reticulum. The troponin tropomyosin complexes again cover their binding sites on the actin, and then the myosin actin binding are now prevented, and the muscle fiber relaxes. Okay, let's talk about some of the energy sources. So you have ATP reserves. It's the first source of energy for muscle contraction. It's the preferred. And muscle cells themselves only store a small amount. Now, creatine phosphate, this is the initial source of energy to regenerate the ATP from the ADP and the phosphate, where the ATP had split. It stores energy in the phosphate bond, so that's what ATP is. There's 3 phosphates. Each bond holds the energy. ATP plus the creatine phosphate can only fuel about 10 seconds of intense muscle contraction, though. So, cellular respiration is really important to fuel the longer periods of muscle contraction. We break down glucose, we produce more ATP, and you remember that from the previous chapter. Glucose is stored as glycogen in muscle cells, though, so we certainly can make use of that, so we can have cellular respiration happening at that level. Okay, when cellular ATP is high, you'll notice that we can regenerate creatine phosphate. But when cellular ATP is low, we have to use some of the creatine phosphate. Okay, the phases of cellular respiration. You have glycolysis at the very beginning. That's the anaerobic phase. It occurs in the cytoplasm. It doesn't produce much ATP at all. Then you have the aerobic phase, which includes citric acid cycle and electron transport. This occurs in the mitochondria, and it produces most of your ATP. Now, myoglobin also stores extra oxygen in your muscles, in addition to the hemoglobin of your blood. Okay, so this shows that whole process of cellular respiration. Here we are inside of a cell. Glucose is broken down to pyruvic acid. This produces a couple of ATP. Then if there's oxygen available, the pyruvic acid can go into the mitochondria, into the citric acid cycle, which produces a little bit of ATP. But ultimately, with all the electrons we're gaining from the initial glucose as it's coming through, we will take all of those electrons, funnel them to the electron transport chain, and then that's where we produce a heck of a lot of ATP. Now, if you don't have enough oxygen, you still can produce a little bit of ATP through the anaerobic phase, but the problem is ultimately lactic acid starts building up as a byproduct of anaerobic respiration, and that will fatigue a muscle. And it's also one of the things that makes you sore. Okay, let's talk about oxygen debt. During rest or moderate exercise, the respiratory and cardiovascular systems will supply enough oxygen to support aerobic respiration. But there is an anaerobic threshold or lactic acid threshold. You have a shift in metabolism from aerobic to anaerobic during really strenuous muscle activity when the above systems cannot supply the necessary oxygen. And that's when the lactic acid builds up. And then oxygen debt occurs. So most of the oxygen in strenuous exercise is used to produce ATP for your muscle contraction, not for converting lactic acid back to glucose. So at the end of strenuous exercise, muscles can be left with oxygen debt. That's the amount of oxygen that's needed by your liver cells to convert the accumulated lactic acid back to glucose and to restore your muscle ATP and the creatine phosphate concentrations. Okay, so this is just a continuous cycle, as we say. And hopefully that oxygen debt will be repaid ultimately as the muscles relax and everything goes back to normal. Muscle fatigue is the inability to contract a muscle, and the common causes of muscle fatigue include decreased blood flow, an ion imbalance across the sarcolemma, loss of desire to continue exercising, accumulation of lactic acid. And this idea is a little bit controversial as to whether that really does cause it or not. There's evidence that it does, but there's some other caveats to it, I guess I should say. Okay, muscle cramp, you've heard of this. Sustained involuntary muscle contractions, you probably have had one of these at some point, can be caused by changes in electrolyte concentration in extracellular fluids in the area. So you always hear people talk about you need to make sure you replenish your electrolytes, and a lot of that's going to be found in things like Gatorade. Okay, heat production. Well, a lot of heat is produced by muscle contraction. So heat is a byproduct of cellular respiration. And muscle cells are the major source of body heat. More than half the energy released in cellular respiration becomes heat. Then less than half is actually transferred to ATP. Blood then transports the heat throughout your body core. All right, so let's talk about the muscular response. So muscle contraction can be observed by removing a single skeletal muscle fiber, like from a frog, and connecting it to a device that senses and records changes in the overall length of the muscle fiber. The electrical stimulator is what's used to promote the contractions. Okay, there is a threshold stimulus. So this is the minimum strength of the stimulation of a muscle fiber that's required to cause contraction. When the strength of the stimulus reaches threshold, then an action potential is generated, and the impulse spreads through the muscle fiber, which releases calcium from the sarcoplasmic reticulum. So we're coming back to that, and it activates the cross bridge formation of the myosin and the actin. One action potential from a motor neuron releases enough acetylcholine to produce threshold stimulus in muscle fibers, causing a muscle impulse. Now, once we get into nervous system, we'll talk a lot more about threshold and action potentials. Okay, a twitch, this is a contractile response of a single muscle fiber to a single impulse. And there are different periods of time associated with a twitch. You have a latent period, which is a delay between the stimulation and the start of the contraction. Then you have the period of the actual contraction. This is where the fiber pulls at the attachments. And then finally, you have a period of relaxation. This is where you have the pulling force decreasing. And this graph shows you those different periods. Latent period, period of contraction, relaxation. Again, this is an individual fiber. The length of the muscle fiber before stimulation will determine the amount of force it is able to develop. So you have what's called an optimum starting length, and that is the resting length of the muscle fiber. It allows the greatest force to develop. So if that muscle is fully relaxed and stretched. So stretched muscle fibers, if they're stretched too much, develop less force, since some myosin heads cannot reach the binding sites on the actin. So you don't want them overstretched. Shortened muscle fibers are a problem also because they develop less force, because they're already in a compressed sarcomere and they can only shorten so much. Okay, so here's what you want. This is the optimal length. So you want to be able to bring these fine fibers, the thin fibers, or fibrils, I should say, or filaments. This would be, again, the actin filaments, and we're wanting to pull those actin filaments, thin filaments, towards each other. And that's being done by these myosin heads. So notice we have a lot of space here to put the two thin filaments together. That's optimal. If it's overly shortened, already contracted, you don't have any more room for contraction to occur. If you're overly stretched, notice that there's a lot of myosin heads that don't even come into contact with the thin filament. Okay, some other terms we use with muscle contraction. Summation, the process by which the force of individual muscle fiber twitches combine when the frequency of stimulation increases. This produces a sustained contraction, and it can lead to partial or complete tetanic contractions. Okay, so again, if you don't need all of those fibers to contract at the same time, they don't. But summation enables you to use many of them together to get a real combined constant contraction. You may have heard the term tetanus before, but when we use the term here, we're not talking about the disease. Anyway, partial tetany, this occurs at higher frequencies of stimulation. So time spent in relaxation between twitches will become very brief in partial tetany. And complete tetany, this occurs at very high frequencies of stimulation. It does not occur in the body, only in the laboratory. You certainly don't want it to happen. It's a forceful, sustained contraction that has no relaxation between the twitches. Okay, this shows you the different levels of contraction and tetanus, and it shows summation ultimately. And so again, if we keep increasing the stimulus so it doesn't have time to fully relax, then we can keep them in a more constant state of tetany. Okay, motor units. A motor neuron plus all the muscle fibers it controls is a motor unit. And a whole muscle consists of many of these motor units. Coarse movements, which are large movements, are produced with large numbers of fibers in a motor unit. So like standing up, so in your legs. Precise movements are produced with fewer muscle fibers in a motor unit, like if you're riding or you're picking something up with your finger. Okay, recruitment, this is an increase in the number of motor units that are activated to produce more force. Certain motor units are activated first, and then others are activated only when the intensity of the stimulus increases, so when you need more motor units. As the intensity of a stimulation increases, recruitment of motor units continues until all the motor units are activated. And this shows those different motor units. So here's motor neuron or motor unit 1, that's a blue one. And notice it's touching this fiber and that fiber and that fiber. So that's all part of motor unit 1. Motor unit 2 are the red ones. Okay, sustained contractions. So smaller motor units, or the smaller diameter axons, are recruited first. The larger motor units with the larger diameter axons are recruited later. Summation and recruitment then can produce sustained contractions of increasing strength. Whole muscle contractions are smooth movements. You may have heard this before, muscle tone or tonus. This is a continuous state of partial contraction in resting muscles. So if we talk about someone being toned, it's because, well, to some degree, it's because their muscles are in a somewhat steady state of some contraction. Isotonic, this is when the muscle contracts and changes length. This means equal force. And the types of isotonic contractions include concentric, where you have shortening contraction that occurs when you're lifting an object, and eccentric, where you have a lengthening contraction. This occurs when the force is not sufficient to lift the object. So it might pull it down. Okay, isometric is muscle contracting but does not change its actual length. So isometric means equal length. So there's tension, but parts that are attached to the muscle do not move. So this is like you pushing against something that doesn't move. Your muscles are definitely contracting, you're pushing, but that object is not moving, maybe because it's too heavy. So whenever you're doing any kind of action that actually moves a part of your body or moves something, then that is isotonic. Okay, so here in this first picture, the muscle contracts with force greater than the resistance and shortens. That's concentric contraction. In the second case, the muscle contracts with force less than resistance and it lengthens. That's eccentric contraction. And then you can see the muscle contracts, but it doesn't change length because you're pulling against something that doesn't move. That's isometric. Muscles do vary in many different ways. One, contraction speed, they can be slow or fast. The way that they produce ATP, we can say it's oxidative or it's glycolytic. There's 3 types of muscle fibers that are found in humans. All muscles contain a combination of the types of fibers. So you have slow twitch fibers, or what we call type 1. They're always oxidative. So they're resistant to fatigue. We say they are red fibers, and it's because they have abundant myoglobin in them, and they have a really good blood supply and many mitochondria. They have slow ATPase activity, so they are slow to contract. So that's your slow twitch fibers, or type 1. Okay, your fast twitch fatigue resistant fibers are type 2A. They're an intermediate twitch fiber. So they're intermediate in their oxidative capacity. They also have an intermediate amount of myoglobin. They're more white fibers. They're still resistant to fatigue, and they have rapid ATPase activity. And then you have the fast twitch glycolytic fibers, or type 2B. These can undergo anaerobic respiration or glycolysis. They are white fibers, so there's less myoglobin. They have a poorer blood supply than the slow twitch fibers. They have fewer mitochondria than the slow twitch. They have more sarcoplasmic reticulum than the slow twitch. They are susceptible to fatigue. And they have fast ATPase activity, so they contract rapidly. All right. Well, using your muscles is very important. You don't want to just let them go. Hypertrophy is when you have enlargement of skeletal muscle, and that happens when you exercise. So hypertrophy is a good thing for your skeletal muscles. You do not want atrophy. So this is a decrease in size and strength of the skeletal muscle that is unused, and certainly does happen to people. Aerobic exercise stimulates slow twitch fibers more. In response, fibers increase their capillaries and their mitochondria. Forceful exercise, though, stimulates mainly the fast twitch fibers. And in response, the fibers produce new actin and myosin filaments, and the muscle enlarges. But it is important to do both aerobic and what we might call anaerobic activity. Okay, so that's your skeletal muscle. Now, as I said, we're going to come back to those individual muscles in the next part of the chapter in my next video. But for now, let's talk about the other types of muscle just briefly. So smooth muscle. Compared to skeletal muscle fibers, smooth muscle fibers are shorter. They have a single centrally located nucleus. And remember, the skeletal muscle is multinucleated. The smooth muscle is elongated with tapering ends. You have myofilaments that are randomly organized. They do not have striations. They lack transverse tubules. And sarcoplasmic reticulum is not very well developed. And that's your smooth muscle. There are different types of smooth muscle, two types. Multi-unit smooth muscle has cells that are less organized. They function in separate units. The fibers function independently. And this is like the iris of the eye and walls of your blood vessels. They are stimulated by neurons and hormones. Okay, visceral smooth muscle. It's a single unit smooth muscle where the cells respond as a unit. Sheets of the spiral-shaped muscle fiber is the organization pattern you see here. The fibers are held together by gap junctions. They exhibit rhythmicity, so they have rhythm. They conduct peristalsis. So you find them in most of your hollow organs, and they like push material through them. It is the more common type of smooth muscle. Smooth muscle contraction does resemble skeletal muscle contraction in that you have an interaction between actin and myosin. They both use calcium and ATP. Both are triggered by membrane impulses. They are different from skeletal muscle contractions in that smooth muscle lacks troponin. It uses calmodulin instead. It has two neurotransmitters that affect smooth muscle. So you have acetylcholine, but you also have norepinephrine. Okay, hormones can stimulate or inhibit smooth muscle. Stretching can trigger smooth muscle contraction. Smooth muscle is slower to contract and relax. And smooth muscle is more resistant to fatigue. Smooth muscle can also change length without changing its tautness. Okay, and the third kind of muscle, cardiac muscle. Cardiac muscle is located only in the heart, and they are striated muscle cells. The muscle fibers are joined together by intercalated discs, and this is one of the ways you can tell it from a skeletal muscle. The fibers branch. They contain a single nucleus. Another way you can tell that it's cardiac muscle. It has a network of fibers that contract as a unit. We call this a syncytium. They are self-exciting, they are rhythmic, and they have a longer refractory period than the skeletal muscle does. They also have no sustained or tetanic contractions. You certainly would not want that to happen with your heart. Okay, the table 9.3 is a good table to look at that compares the different types of muscle tissues. Okay, and we will, let's see. Yeah, before I'll go ahead and cover this very quickly also before we get into the individual muscles. So 9.7, skeletal muscle actions. So skeletal muscles will generate a lot of different types of body movements. Action of muscle depend mainly on these two factors, the type of joint that it's associated with, and the way the muscle is attached on both sides of the joint. So when the bones or the body parts move, bones and muscles will act as levers. And there's four basic components of levers. You have the rigid bar or the rod, and that's basically the bone. You have a fulcrum or the pivot point on which the bar moves, that's your joint. The object moved against resistance, that's the weight. And then the force that supplies the energy for movement is the muscle. So that's the four parts of the lever system. Bending and straightening of the upper limb at the elbow is an example of lever action of bones and muscles. During bending, the forearm bones act as the rigid bar. The elbow joint acts as the fulcrum. The hand moves against the resistance provided by a weight, and this represents what we call a third-class lever. It's arranged in a resistance, force, fulcrum sequence. Okay, but there are other kinds of levers. The first one we talk about, the first class, you have the resistant, well, let's see. Yeah, same thing we were just talking about. So we have the resistance here, fulcrum in the middle, force here. And they're using scissors as an example here, really. But the movement of the arm probably was a more specific or better example. Now, you also have in your body second-class levers. In this case, they compare it to like the way a wheelbarrow works. You have the fulcrum at the end, the resistance in the middle, and then the force at the other end. And then the third-class lever, you have the resistance at one end, but now you have the force in the middle and the fulcrum at the other end. And they compared this to using tweezers. Okay, now this is that basic lever we were just talking about. You have flexion and extension at the elbow. So in this case, you have the fulcrum in the middle, the resistance at the end. But notice in this particular case, though, because the muscle is attached to your radius, the force now is actually between the fulcrum and the resistance, really. Okay, and this other one shows the opposite, because now if you are extending your arm, you're going to use a triceps on the back, okay? So in this case, the muscle pulls on the ulna here this way. So now we have the force, and we have the fulcrum in the middle, and we have the resistance at the end. Okay, now how bone moves is dependent on where the muscle is attached to it. So one end of the skeletal muscle is more fixed, the other end is movable. We call the movable end the insertion and the less movable end the origin. So when a muscle contracts, insertion is pulled towards the origin. A muscle can have more than one origin or insertion. Okay, so here you see the insertion of the biceps brachii is right here on the radius. Okay, the origin is up here at the coracoid process of the scapula. Most skeletal muscles function in groups. So the roles of the muscles in performing certain actions include an agonist. That's the muscle that causes an action. A prime mover. The agonist primarily are responsible for the movement. In some cases, the term agonist and prime mover are used interchangeably, and they are most of the time. We usually can use either one of those terms. The synergist is the muscle that assists the agonist and prime mover. The antagonist is the muscle whose contraction causes movement in the opposite direction of the prime mover. Okay, and there is a sheet of muscles to know that you need to start memorizing. You see many of them here, and also the posterior part as well, or dorsal. This is where I will stop this particular video, and we will proceed at the next video going through each of the major muscles.